Hello everyone and welcome to Pocket Size Bio, Dr. Casco. In today's video, we're going to go over Campbell Biology Textbook, Chapter 9, which is Cellular Respiration and Fermentation. This chapter is broken down into 6 subsections that I'm going to break the whole chapter into 2 videos. In this video, we're going to go over 9.1 to 9.3, talking about catabolic pathways, glycolysis, and pyruvate oxidation. So let's get to it. Let me just move on to the next little slide here. Okay, so section 9.1 is catabolic pathways yield energy by oxidizing organic fuels. As we know, living cells and living systems require energy from outside sources. Okay. We need energy to assemble polymers, to pump substances across the membrane, active transport, to move around, to reproduce. Ultimately, everything we do requires energy. In the case of animals, we need an outside source of energy, which is food. And you can see here, figure 9.1. Let me just zoom into this. Hey, you have this animal here, the hoary marmot, Marmota caligata, obtains energy from its cells by feeding on plants. In the process of cellular respiration, mitochondria in the cells of animal, plants, and other organisms break down organic molecules, generating ATP and waste products, carbon dioxide, water, and heat. Note that energy flows one way, but chemicals can be recycled or are recycled. So one of the main things we're going to see in this chapter, how is the chemical energy stored in food used to generate ATP? And as we know already, ATP is the molecule that ultimately drives a lot of the cellular work that is occurring in the cell, in the organism. We have this little diagram here. We're getting light energy as a source of photosynthesis, to drive photosynthesis, which ultimately generates organic molecules like glucose and oxygen, which is used by other organisms like animals to undergo the process of cellular respiration, where they break down glucose, oxidize glucose to produce ATP and heat as a byproduct, whilst all of this is occurring in the mitochondria. The process of cellular respiration outputs carbon dioxide and water, which is then again used in the process of photosynthesis. So they do go hand in hand, and we're going to see this relationship once we finish photosynthesis. Okay, let me go back to my notes. Zoom in here. Okay, as I mentioned already, photosynthesis generates oxygen and organic molecules, and that is used as fuel, okay, for cellular respiration in mitochondria. Cell respiration is broken down into 3 key pathways, okay? Glycolysis, the citric acid cycle, and oxidative phosphorylation. We're going to see those in depth in this chapter. Some information that we need to know a little bit more in detail: catabolic pathways and the production of ATP, right? Organic compounds in itself, they carry potential energy, right? And forms of potential energy can be found in the bonds and the electrons, how the arrangement of electrons are. So having this potential energy, they can serve as fuels, okay? And one process of catabolic pathways is fermentation, right? It's a relatively simple pathway where you're using glycolysis, okay, ultimately to, degrade sugar, to extract some energy from sugar to a certain extent. There's no use of oxygen in fermentation, and this is a form of anaerobic respiration. Anaerobic, meaning that there's no involvement of oxygen. Okay, but the output of energy isn't the greatest, if I can say, right? There's a more effective catabolic pathway, which is aerobic respiration, and most eukaryotic organisms do this process as it yields a higher amount of ATP. Okay, so the term cellular respiration, it includes both aerobic and anaerobic processes, but generally the term cellular respiration commonly refers, and in the book included, as an aerobic process, meaning that it's going to use oxygen. Okay, and this whole process of cellular respiration is similar in the principle with combustion of gasoline, right? Where you have food as a fuel source, and that fuel source ultimately will burn to be extracted, for energy to be extracted, and the output or the exhaust is carbon dioxide in water. In this case, it's like a slow burn, right? And you can there's different types of molecules that can be used as fuel. You have carbohydrates, you have fats, and proteins can be used as fuel. And we focus on, of course, glucose in cellular respiration, as that is what is being used, right? But the catabolism of glucose is exergonic. It's an exergonic process where delta G, that free energy change, is negative, 686 kilocal per mole. And this is a number I do recommend remembering. Okay. And that energy is then used to produce ATP for cellular work. Ultimately, this whole idea of cellular respiration is for the whole purpose, of synthesizing ATP and ATP. And we have this little diagram here, as we're going to see later on in detail, to work on this complex ATP synthase, okay, to process this molecular machinery for this protein to actually move and harness the potential energy of a hydrogen plus ion gradient, extract that potential energy to produce ATP from ADP and AADP and inorganic phosphate. So this is kind of our net goal here, ATP production. So let's go into more detail in the next little panel here. All right. Before going into cell respiration, we do need to talk about some concepts, mainly redox or redox reactions. So you have oxidation and reduction reactions. Okay, and redox reactions, as you probably have seen in chemistry class, they release energy when electrons move closer to electronegative atoms. Okay, they release energy when electrons move closer to electronegative atoms. Catabolic pathways, they transfer electrons from food, releasing energy to synthesize ATP. And these redox reactions, you have the same concept. Okay, there's a transfer of electrons from one reactant to another. Okay, and some concepts that you need to familiarize yourself: oxidation, reduction. What is referred to as oxidation is any substance that has lost electrons. Okay, they have oxidized. Reduction is the addition of electrons to another substance. Okay, so you say that those substances have become reduced. Okay, so adding electrons is called reduction because it reduces the atom's positive charge. Okay, and related terms here, we have this little diagram. So X, the electron donor, is the reducing agent. Okay, so you have this reaction here. Any X molecule will provide the electron to the other substance. So it's the reducing agent. It's the agent that will provide a reduction to the other molecule. And then the Y substance, it's the electron acceptor. It is referred to as the oxidizing agent. It is the one that will receive the electron from the other substance. So one becomes oxidized and the other one becomes reduced. An example reaction here, very simple, not necessarily biology related, where you have sodium, and sodium will provide an electron to a chlorine atom, where you have this sodium plus ion and chloride ions here. Okay? Oxidation, reduction. And we're going to see this in multiple instances where during the whole cellular respiration processes. And in redox reactions, you always require a donor and an acceptor. Somewhere, some other substance must receive the electron. Okay? So you're undergoing this constantly in biological systems in particular. Let me just zoom in now. Okay, here's another example of methane and in methane combustion. So redox reactions can also occur when you're having a change in the degree of sharing of electrons within covalent bonds. So you don't necessarily have to detach an electron completely, but you're getting just an unequal sharing of electrons. Okay. So in methane combustion, for example, you have non-polar bonds convert to polar covalent bonds in this particular reaction. Okay, just to go through the notes before I get into this panel here, when methane reacts with oxygen, electrons end up farther from carbon and closer to oxygen. Okay. When methane reacts with oxygen, okay, electrons end up farther from carbon and closer to oxygen. Okay, so if we take this example here, you have methane reacts with oxygen to produce carbon dioxide plus energy plus water. So in the product of methane, ultimately yielding carbon dioxide, you will have this unequal distribution. So the electrons will be closer to the oxygen. Okay? So this is what it's referring here. The carbon atom has partially lost its shared electrons as methane has been oxidized. Let me just move down a little bit here. So the two atoms of oxygen, they share electrons equally, as you see here, okay, in red. They both have the same electronegative strength, so there's an equal sharing of electrons. But when oxygen reacts with hydrogen to form water, and you have this same instance here, right? Electrons are drawn closer to oxygen, as a similar case here. Okay, so you have these two situations occurring. Each oxygen atom has partially gained electrons. Thus, the oxygen has been reduced. Okay, so oxygen, as we know, it's a very electronegative and is one of the most potent. It's the strongest oxidizing agents because it pulls electrons towards itself. So it's become reduced. Okay, and then in order for that to occur, and in order for electrons to move at all, you need to have some energy from somewhere. Right, so energy must be added to pull an electron away from an atom. And so there's always a play of energy within these redox reactions. Okay, again, energy doesn't come from spontaneously. So the more electronegative the atom, the more energy is required to remove that electron. So whenever you're adding some source of energy, a redox reaction can relocate electrons closer to the oxygen, like burning methane. And whenever you burn the methane, it will release chemical energy. Okay? Ultimately combusting. All right? So moving on to a more kind of organic fuel scenario. Right? So you have oxidation of organic fuels or organic fuel molecules during cellular respiration. Again, respiration is a redox process. Glucose gets oxidized and oxygen gets reduced. Okay? We're going to break down glucose into carbon dioxide ultimately. So as this process is going along, and there's many pathways in interplay here, electrons will lose potential energy along the way. And as they're losing potential energy along the way, they're releasing energy. Energy is being released. Okay, so organic molecules in themselves, they are abundant in hydrogen, right? Carbohydrate, a molecule. So they're excellent source of fuel. Okay, so because of this, they have their bonds are a source of hilltop electrons, right? And then these electrons will will release energy as they fall to oxygen. Okay, so they'll release energy. And let me just move this a little bit down here as an example, of course, of cell respiration. Glucose gets oxidized into carbon dioxide. Oxygen gets reduced into water, and there's a tremendous amount of energy that is being released. Okay. Again, so glucose oxidation transfer electrons to a lower energy state, ultimately to carbon dioxide. In each step of the way, as glucose is being broken down, it's releasing energy for ATP synthesis. And again, carbohydrates and fats could be used, are good reservoirs for this idea of electrons and releasing energy and being associated with hydrogen. Okay, so in general, these molecules, these hydrocarbon, sorry, I mentioned carbon, hydrogen, maybe here, hydrocarbons. Yeah, they're very stable because of the barrier of activation energy. Right? Remember, activation energy. You need a certain amount of energy for the reaction to proceed. If you didn't have this energy of activation, glucose and other organic molecules will essentially combust immediately with oxygen, and we don't want that, right? Or else everything will catch on fire. Okay, so but if you have an initial source of energy, if ignited, right, glucose can burn to release 686 kilocalorie per mole of heat. Okay. Again, this number. Remember this number per molecule of glucose. So this reaction cannot happen at body temperatures, fortunately. Okay, or else we'll be combusting. Enzymes lower the activation energy, right, allowing oxidation in a series of steps. So this is important because you can release safely, securely, little bits of energy to produce ATP and ATP and ATP. Okay, so these are referred to as a kind of a stepwise method. Right, so you want to harvest energy, and we're going to see more specifically how that works. And one of the major players to harvest that energy of ATP synthesis is NAD plus, okay, or NAD plus, within the electron transport chain. Okay, so cellular respiration does not oxidize glucose in a single step, as I mentioned before. It is broken down in a series of steps, catalyzed by many enzymes during the process. And as these steps go along, electrons are stripped from glucose. Okay, so you have in many oxidation reactions, the electron is transferred with a proton as a hydrogen atom. So basically, electrons and hydrogens are being played around here. Okay, and in order for this to happen, ultimately in the electron transport chain, you need a molecule to be passing down, to be transporting these electrons and these hydrogens from one place or from one reaction to another, from one complex to another. And this is the kind of the go-to structure here. It's called NAD plus or NAD plus. It's nicotinamide, nicotinamide adenine dinucleotide, or NAD. Okay, and then you have here its structure NAD plus. Here's the nicotinamide in its oxidized form. You have this nitrogen plus atom there. That it can cycle easily between an oxidized state and a reduced state. So you have NAD plus cycles easily between its current form and its reduced state or form, which is NADH. Okay, so NAD plus functions, remember, as an oxidizing agent. It means it's going to pull the electron and collect electrons and hydrogens from somewhere else to reduce, to become reduced. Okay, let's continue here. All right, so just to zoom in a little bit here. So there's going to be several enzymes that you need to study. Dehydrogenases, okay, are critical enzymes to memorize the name. They strip hydrogen atoms from a substrate, so they oxidize the substrate, and then ultimately they'll pass these hydrogens and these electrons to NAD plus. Okay. Moreover, they'll pass 2 electrons and 1 proton, 1 hydrogen, to NAD plus. The other proton is released as a hydrogen plus ion into the solution. Okay, so by receiving 2 electrons and 1 proton, 1 hydrogen, NAD is charged. NAD's charge is neutralized, becoming NADH. Okay, so NAD plus is an oxidizing agent, as I mentioned before, and you're going to see this in many steps, particularly in the Krebs cycle, as we go down the glucose breakdown. Okay, electrons carried by NADH, they lose very little of their potential energy, so they're kind of chaperoned down to the electron transport chain, as we're going to see later on. Okay, ultimately helping ATP being synthesized. This is a visual representation of what we're seeing here. You're getting 2 hydrogens from a organic molecule source. Dehydrogenase will take out these electrons and these hydrogens to reduce NAD plus to NADH. Okay, and then these one of the hydrogen plus ions will be in solution. Okay, good. All right, getting a little bit now into electron transport chain. Now, just to understand in essence what it is, and then we're going to see it in more detail in the mitochondria. So you have this system called an electron transport chain to essentially break the fall of electrons and pass these electrons from one molecule to another, to another, to another. And what happens here, right, is that this electron transport chain, it consists of a series of molecules. They're represented by these little spinners, if I can say, turbines, if I can say, right? And we're going to see it, again, of course, the actual electron transport chain that is built inside the inner mitochondrial matrix, okay, for eukaryotic cells. And for prokaryotic cells, it's embedded within the plasma membrane, okay? And the electrons, that you obtain, that you harness from food, they'll be passed down to NADH as kind of carriers. They're actually electron carriers. And they're passed down to the top of this electron transport chain, which then gets shuttled from one molecule to another, to another, to another, ultimately to oxygen, which then becomes water. Okay? As we're going to see a little bit here. So electron transfer from NADH to oxygen is an exergonic process. Okay, so it's generally a spontaneous process with a delta G of negative 53 kilocalories per mole. So as electrons are being passed through each molecule is actually more electronegative. Okay, in itself, these molecules are more electronegative, ultimately to the last acceptor of the electron, which is oxygen, the most electronegative molecule there that you see there. Okay, until they reduce oxygen, and then each carrier is more electronegative than its uphill neighbor. So let's say if you're seeing this little panel here, an electron is passing down from one molecule to another more electronegative molecule, and so forth, and so forth, and so forth, until you pass to oxygen, which produces water. Each time you're passing down these electrons, you have a source of energy to produce ATP. Okay, and this has to do with hydrogen pumping throughout the membrane, which we're going to see a little bit later. Okay, so it's a more controlled way of extracting the energy from glucose. If you were to do this in one blow, hydrogen plus oxygen reacting together, that whole combustion will have an incredible release of energy, an explosive release of heat and light energy, an explosion. So you definitely don't want that to occur in your body. Instead, you have this system that is more efficient within the cell. Okay, so ultimately, what happens here, you have electrons jumping from glucose. It'll go from glucose to NADH, to the electron transport chain, to oxygen. As this is passing down, you are liberating energy, right? And you're harnessing energy to produce ATP. Okay, let's go on to the next page here. So the stages now of cell respiration, getting into the idea of redox reactions and the electron transport chain, we now get into the concept of cell respiration. So it's broken down into three parts in general, right? Glycolysis, pyruvate oxidation, and the citric acid cycle, okay? And oxidative phosphorylation, okay? There's three main stages, though. Here you see two substages. Here, pyruvate oxidation and citric acid cycle, but it's considered as stage number 2. Okay, so you have glycolysis, citric acid cycle, and oxidative phosphorylation. Again, in terms of aerobic respiration and cellular respiration, it's focused more on the citric acid cycle and the electron transport chain oxidative phosphorylation. So stages 2 and 3. Okay, glycolysis is included because it produces that starting material to be used in the other stages, essentially pyruvate, but you don't use oxygen in itself. Okay, so glycolysis, and we're going to take a look at each of them more in detail as we go along. Okay, glycolysis occurs in the cytosol, and ultimately it breaks down glucose into 2 pyruvate molecules. Okay, in eukaryotic cells, and again, we're going to see this in more detail, pyruvate, so this molecule that is being produced, is kind of further groomed, changed in the mitochondria, and it's oxidized into what is called acetyl-CoA. Okay, before it enters the citric acid cycle. There's several steps in glycolysis and in the citric acid cycle, ultimately producing NADH. Okay, some steps do produce ATP also, but we will see the majority of ATP production in oxidative phosphorylation. Okay, and you can see a general overview as what I'm referring to here. Glycolysis, it is important to know how much ATP is being produced in each particular stage and where the stages are located. Okay, glycolysis occurs in the cytosol, pyruvate oxidation and citric acid cycle occur in the mitochondria. okay, and also oxidative phosphorylation that occurs on the membrane, inner membrane. Okay, so this is a very good diagram for you to kind of be able to summarize for yourself, to explain to yourself. Okay. All right. So let's continue on. In stage 3, the electron transport chain, yes, it does accept electrons from the citric acid cycle and the previous stages, essentially, where NADH is being formed, and it's accepting all those NADHs, collecting their electrons. Okay, as I mentioned before with the electron transport chain, you want to have those electrons to ultimately form water and extract all that energy as it's going down that cascade, that chain. Okay. We're going to take a look at other forms of ATP production or substrate or phosphorylation. So this formation of ATP synthesis is called oxidative phosphorylation, ultimately driven by redox reactions. Okay, as I mentioned before, in eukaryotic cells, it occurs in the inner mitochondrial membrane. In prokaryotes, it's happening in the plasma membrane. Okay, this last step accounts for almost 90% of ATP production. So it's the one that harnesses the majority of the energy and produces the majority of ATP. Okay, between 30 to 32 ATP molecules per one molecule of glucose. Again, this is also something you should memorize, remember. Okay, some ATP can be produced in a lower extent by what is called substrate-level phosphorylation, and this is just the interplay between enzymes and substrates that have a phosphate group. They can attach together, making an enzyme complex, substrate complex, and they can pass this phosphate to a DP to produce a TP. Okay, so you don't necessarily have to go through the entire process of cell respiration. Just by this substrate level phosphorylation, you can produce a certain amount of ATP. Of course, not as much as the electron transport chain. Okay, but just keep that in mind as we're going to go along. We're going to see this occurring. Okay, good. So this is just a very, very brief overview of cellular respiration, aerobic cellular respiration. We're going to go into glycolysis now. Okay. Let me zoom in a little bit here. So 9.2, this is glycolysis harvests chemical energy by oxidizing glucose to pyruvate. That's the main thing you need to know here. Okay, the formation of pyruvate. Okay, so during glycolysis, remember you have glucose. Glucose is a 6-carbon compound, 6 carbons. So you're going to split it in half into a 3-carbon sugar. That's the main goal we're gonna see there. Okay, and this you must know, pyruvate, this molecule pyruvate. And within this whole process that is occurring in the cytosol, okay, you're having many enzymes working together, breaking down glucose and changing the structure. And as it's going along, changing the structure of glucose, you're going to have 2 molecules of pyruvate that are 3-carbon compounds that have 3 carbons. Generally, glycolysis is broken down into 2 parts: an energy investment phase. So you will need to be using a little bit of ATP to be breaking down glucose and to be creating isomers in some cases of glucose. And then you're going to have an energy payoff phase. Okay. Again, energy investment, you're going to use ATP. Energy payoff, you're going to produce ATP along with NADH. Okay, so that's something you do need to know. Okay. All right. So something important to note, glycolysis can occur whether or not oxygen is present. It's independent of oxygen. And we're going to see that more with fermentation. So keep that in mind. They might, tests might ask you which reactions occur, which processes occur without or with the presence of oxygen. So keep that in mind. Okay. Glycolysis can occur with or without oxygen. Okay. If oxygen is present, then the energy in pyruvate and NADH can be extracted by pyruvate oxidation and the citric acid cycle and phosphorylation, oxidative phosphorylation, basically the mitochondria. If oxygen is not present, then glycolysis will occur and then followed by fermentation, as we're going to see in the later sections there. Okay. Just a brief summary of glycolysis. This probably you do need to memorize, learn the amounts of ATP being used, the outputs, energy payoff phase. You're using 4 ADP to produce 4 ATP, okay? And 2 NADHs are being produced, plus 2 pyruvates. I think moreover is the net, okay? So make sure you learn the net inputs and outputs, okay? But understand when the energy investment part is happening and whenever the energy payoff phase is happening. Okay, but this particular diagram here is the most essential to know. Okay, so if you're an AP student, you probably won't need to know all these intermediates here that you see, glucose 6-phosphate, fructose 6-phosphate. But if you're an undergrad, you probably do need to know. I'm going to go along and we're just going to read through these so you can get a sense of what is happening. But again, AP students probably aren't required to know every single detail of the Krebs cycle. Okay, so the specific steps of glycolysis. Remember, the whole purpose of cell respiration is to extract energy from glucose. Oxidize glucose slowly, okay, into smaller, breaking it down slowly to extract as much electrons and reduce NAD plus to NADH. Okay, so let's take a look. We have glucose here, the starting compound. You're getting some enzymes, of course, throughout every single step. And what happens is that ATP is being used to create, to enable glucose to become more chemically reactive. So a charged phosphate also traps the sugar in the cell, so it keeps it from going anywhere else. And then if it's chemically more reactive, having this phosphate here, it turns into glucose 6-phosphate. Basically, that you have a phosphate group on the 6th carbon. That's all there is. Then you have another enzyme, phosphoglucoisomerase. This just converts into an isomer of this compound. And then here, you're investing a little bit more energy, okay, using phosphofructokinase to transfer a phosphate group from the ATP to the opposite side of the molecule. So you have fructose 1,6 biphosphate. Now, all that means is that you have 2 phosphates being attached to the 6-carbon molecule now. So I'm going to refer to it as a 6-carbon molecule, as it's generally referred to this way. Okay? Fructose 1,6 biphosphate. Okay? So this aldolase, maybe this one you need to know. Even AP students, I do recommend knowing this particular enzyme here, which will cleave the molecule in half, break the molecule in half, into 2 3-carbon compounds. So now instead of having one long 6-carbon compound, you're going to have a molecule that is 3 carbons. Okay? Glyceraldehyde 3-phosphate, G3P. So this one also I do recommend remembering. Okay? So we have glucose. Glucose will break down into a molecule called G3P, glyceraldehyde 3-phosphate. Okay? And we're going to continue on to the next step here. Okay. So if you have the textbook, this is connected into one like this. Okay, but just to zoom in a little bit better here. So you have this G3P, and this G3P will, in a sequence of reactions, G3P is going to be oxidized. Okay, and then you have this, again, enzyme dehydrogenase, okay, which will remove the electrons and the hydrogens, as we talked about already, into your NADH. Okay, so here is good to know that you're synthesizing NADH. Okay, and then this will produce 1,3-biphosphoglycerate. Okay, and here you're getting the energy payoff phase. Okay, always occurring in redox reactions. Okay, so this would be a very high energy product. This high energy product will yield, provides the energy to give ADP, a phosphate, and produce ATP, phosphoglycerol-kinase. Okay, and then you'll have your first ATP being produced. Okay, the phosphate group is transferred to ADP, and this is important to note that you are producing ATP by substrate-level phosphorylation, not the electron transport chain, substrate-level phosphorylation. Okay, so the enzymes are working here to produce ATP. This will produce 3-phosphoglycerate. You're getting other enzymes here, phosphoglycerol mutase. This will just relocate the phosphate groups. Okay, producing an isomer here, 2-phosphoglycerate. Again, you'll have another enzyme, enolase, will cause a double bond to form as you're removing water. Okay, so here's where you're seeing water being formed, released, an output. Okay, extracting water molecules. Okay, so if they ask you, you can be asked, where's water being released? You can say in glycolysis, when you're forming the pyruvate in the energy payoff phase, for example. Okay, that being formed, you have another kind of intermediate molecule, phosphoenol pyruvate, and then still this has some energy that could be harvested, which is ultimately produced in ATP and given to ADP to produce ATP, and then you get pyruvate. Okay, this is your last molecule. Remember, this is happening twice because you have 2 3-carbon molecules. So if you see there's a little page in the back, this is happening twice. Okay, but of course, they already put 2 and 2 for you to know already. Okay, this is a net output. Since it's occurring twice, you produce 2 NADHs, you produce 2 ATPs, and 2 water molecules, and another 2 ATPs. Okay? So I want to go back here just to give you a sense of what is happening here. The net input and outputs. Net inputs, you have glucose. And when they produce pyruvate, right? Remember, you are producing 4 ATPs, but in the energy investment, you need to use 2 ATPs. So there's a net output of 2. Of course, 4 minus 2 equals 2. Simple. Okay. And then you have these inputs here, which ultimately reduce NADH and hydrogen plus ions in solution. So that's it for glycolysis. Again, be careful for what you need to learn. Tell your instructor if you're an undergrad or if you're an AP, if you need to memorize all these intermediate molecules. Okay, we're going to go to 9.3. After pyruvate is oxidized, the citric acid cycle completes the energy-yielding oxidation of organic molecules. Okay, it says more than 3/4 of glucose energy is still in the 2 pyruvate molecules, so it hasn't been fully oxidized yet. And again, if oxygen is present and you're an eukaryote, the pyruvate will go into the mitochondria, okay, for oxidation in the citric acid cycle. But if you're a prokaryote, this will be happening in the cytosol, okay? There is no organelles. All right, but we're going to be focusing primarily on eukaryotic systems. So oxidation of pyruvate to acetyl-CoA. All right, so this is, before going into the Krebs cycle, this is another phase that in which glucose is being broken down further into. More specifically, pyruvate now is going to be broken down further. Okay, so pyruvate is going to be converted into acetyl coenzyme A, or acetyl-CoA for short. And this is a multi-enzyme complex that will attach to what's going to be a 2-carbon compound that we're going to take a look. The main things that occur here, and this is the general reaction diagram of what is happening. A carboxyl group is removed as carbon dioxide. Okay, so if you see here, you have the pyruvate 3-carbon compound. Okay, you have your carboxyl group here. Okay, so as it enters the mitochondria, this pyruvate will get oxidized and it will be removed. A carbon molecule will be removed via an enzyme, pyruvate dehydrogenase, okay, which will do this entire process. Carbon dioxide is removed. NADH will be formed, okay, NAD plus will be reduced by NADH. As the carbon dioxide is oxidized and liberated, NADH is formed, and you will have the attachment of a complex called coenzyme A. So then you have this formation called acetyl-CoA, 2-carbon compound associated with this enzyme complex. Okay. Now here is something to notice. This is the first carbon dioxide that is formed from a glucose molecule. Okay, and remember that you have 2 pyruvates. Okay, so you'll have 2 acetyl-CoAs. So 2 carbon dioxides will be removed. Okay, so keep that in mind here. Make sure you're understanding the play between the 2 molecules, right? So due to a CoA group, right, from a B vitamin, so acetyl-CoA has high potential energy. Just note that this acetyl-CoA has good potential energy. So the reaction of acetyl-CoA to lower energy products will be highly exergonic. So we can extract a lot of energy from them. Okay, so acetyl-CoA then will go into now the citric acid cycle, okay, for further oxidation. This is a general representation of what is happening here in the citric acid cycle. Okay, the citric acid cycle, depending on your textbooks, depending on tests and things like that, note that it could also be called the Krebs cycle or the tricarboxylic acid cycle. So if you see these terms, make sure you're familiar with them. Okay, what happens here? Again, there's further oxidation of this organic fuel from pyruvate and so forth. Here, in this case, you're going to have 3 carbon dioxide molecules being released from the pyruvate, including the acetyl-CoA in total, right? So literally in the citric acid cycle, you're going to have 2 carbon dioxides. Okay, so what is happening here is that you're getting intermediate molecules that are coming along and attaching to this acetyl-CoA, changing into isomers, changing into different structures. As they're doing that, they are releasing electrons to be received by NAD plus and be reduced into NADH. You'll have another player, as we're going to take a look, called FADH, okay, which serves a similar function to be an electron carrier. And we're going to take a look at that also. You're also going to produce ATP. ATP, again, by substrate-level phosphorylation. Enzyme phosphorylation. You're going to get these two electron carriers ultimately. A good amount of electron carriers are produced here. That's kind of one of the main idea that you need to know. Citric acid cycle or Krebs cycle, you're having a lot of production of NADH. Okay? And then those will go to the electron transport chain. So in summary, what's happening here, you're having your pyruvate, 3 carbon molecules. Remember, in pyruvate oxidation, 1 carbon dioxide is released, 1 carbon is released, so you have a 2-carbon compound here. And then in the citric acid cycle, this compound is further oxidized, and here's where you have your production of carbon dioxide. Okay, so a question that you can be asked in a test, where does carbon dioxide form in this whole process of aerobic cellular respiration? Right? You can say pyruvate oxidation and the citric acid cycle. And then here, glucose is completely broken down. Okay? And ultimately, what happens? You have acetyl-CoA coming in. As these carbons are removed, electrons and hydrogens are also extracted to produce NADH and ATP. Again, this is happening in the mitochondria. Let's look at it more in detail. Okay. So this is the general gist. Okay. So there's 8 steps. Depending if you're in the AP program or if you're an undergrad, make sure you ask if you are required to know all these intermediate molecules. Okay. But this is one thing everyone should know, that the acetyl-CoA starts this cycle. And the reason why it's a cycle is because the acetyl-CoA enters and binds to a 4-carbon molecule called oxaloacetate. This you do need to know. Okay? You should remember. When these two join together, they produce a compound called citrate. And this is the ionized form of citric acid. That's why it's called the citric acid cycle. Okay? And you're getting a series of intermediate, structures. And again, you're going to see how some carbons are being liberated as carbon dioxide. Okay? So I'm going to kind of zoom into this whole diagram here, and then I'll go through the notes as we go along. I want you to visually see what is happening. So you have oxaloacetate with acetyl-CoA. They bind together, and again, you're having enzymes that are integrated in this whole procedure. But this might be beyond the scope of general bio for undergraduates. This is biochemistry level to know that level of detail. Anyways, so these two bind together. They release the coenzyme A, and you have the 6-carbon molecule. Okay, 1, 2, 3, 4, 5, 6. All right. This is called citrate. During this process of the citric acid cycle, water is released. It's converted into a different, I don't want to say isomer, but yeah, an isomer here. Water is also added just to kind of restructure the molecule. Okay, so you have isocitrate. Okay. And then here is your kind of first major procedure here. Isocitrate is oxidized. Okay. Oxidized, so that means it's going to reduce NAD plus into NADH. Okay. As it's also oxidized, it's liberating a carbon dioxide molecule. Okay. So here's your big loss of energy extraction, your energy extraction here. This turns into a molecule called alpha ketoglutarate. Look at the carbon numbers. You have 1, 2, 3, 4, 5 carbons. Okay. And then when it turns into this, you get another carbon dioxide being removed. Okay. Again, there's a lot of interplay between the structures of these intermediates. ultimately you want electrons to be removed from these molecules. Carbons are also being removed as electrons are being liberated and placed into NADH. Carbon dioxide is released here, okay? And you get NADH. As you get this molecule, now you have a 4-carbon molecule called succinyl-CoA. Yes, this coenzyme complex does come back here, but it's liberated again, okay, into the solution. This molecule is carrying some good amount of energy, okay? So the CoA is displaced by a phosphate group, so it's also released again, and that release provides energy for GTP. GTP is very similar to ADP. It's an energy carrying molecule also, which gets phosphorylized into GTP. And then this GTP ultimately produces ATP. Will kind of carry energy from ADP to produce to ADP to produce ATP. Okay? And then this, again, as I was saying, GTP can also be used to generate ATP. And then you get another intermediate molecule here, succinate. Okay? Since they're still carrying some good amount of potential energy in the electrons, they pass electrons to FAD, reduce it to FADH2. Okay? 2 hydrogens are transferred to FAD, and the molecule gets oxidized into another compound called fumarate. This fumarate will then get hydrolyzed with water, or the molecules will restructure. Hydrolysis, they'll break the double bond here, and you have this next intermediate molecule called malate. This malate further gives the last kind of set of electrons to NADH, NAD plus to produce NADH, and then you go back into oxaloacetate. Okay? The main thing you need to understand here in these cycles is that you use these molecules to come in, gather acetyl-CoA, extract the electrons and the carbons to rally them to pass the electrons to NADH. And then as that happens, they ultimately release these 2 carbons and they regenerate the original molecule, which is oxaloacetate. Okay, so that's why it's called a cycle. All right, so just to go through the notes here to make sure I'm not missing anything. Yes, so you want to start with oxaloacetate, right? Each acetyl group, 3 NAD pluses are made. So what you see here in this particular diagram, okay, you have 1 ADH, NADH, 2 ADH, and the third NADH being formed. You're also producing ATP and FADH2. Now remember, because it's occurring twice, you add them, of course, you multiply them times 2 because you have 2 acetyl-CoAs. So you have 6 NADHs occurring, being produced, 2 FADHs, and 2 ATPs. Make sure you know the numbers, right? It's very important. All right. But here in the context of this page in the book, they're mentioning at this part 3 NADHs to produce this, the formation of FAD. Okay. Now, depending on some cells and tissues and the organisms, you'll get the formation of ATP by substrate-level phosphorylation. Okay. In many animal tissues, GTP is formed, but then ultimately ATP is formed. Okay. Again, yes, this step is only ATP generated directly by the citric acid cycle. This is the key thing that you do need to know, okay? Total yield per glucose from the citric acid cycle is 6 NADHs, 2 FADH2, and 2 ATPs. And then these will be used to go into the electron transport chain. Okay? Sorry about that background noise there. Okay. So that's it for this first half of chapter 9 video. I will produce the next chapter soon. So thank you for watching. The next section will be 9.4. During oxidative phosphorylation, chemiosmosis couples electron transport to ATP synthesis. Okay, hope this video was useful, and I'll see you in the next one. Thank you.